Okay, so chapter 24, we're going to talk about the urinary system. I like to think of the urinary system as basically just a big filtering system. And in fact, all right, we actually give a name to the fluid that is getting filtered from the blood. You know, the first name that we give it is called filtrate. So I just think of it as a big filtering system in which we're going to kind of filter out the blood, keep what the body needs, wants, and then get rid of the stuff that it doesn't need or it doesn't want. So basically, we're going to talk about how that process works, where we're going to convert that filtrate into urine, what we essentially consider the waste, the fluid waste of our body. So a couple of the functions of the urinary system is, like I just said, you know, we're going to get rid of some of the metabolic wastes that occur throughout the tissues in our body, right? It is big, and we're going to talk about all these, how all these different processes are regulated and how these functions occur, all right? But of course, all right, ion levels, and we've learned that ion levels, all right, having the appropriate ion levels are crucial to homeostasis, to the normal functioning of our body in regards to, for example, when we're talking about the neurons. Okay, the neurons have to have a certain amount of sodium, a certain amount of potassium, right, to generate that action potential, that proper ion flow across those voltage gated channels to conduct an action potential. So obviously the regulation of specific ion levels, all right, calcium, we saw the value of calcium in several functions and several systems. All right, we need calcium for muscle contractions, right, and that includes, all right, skeletal, muscle, it also includes smooth and cardiac. Right? We also know that calcium is needed for the hydroxyapatite, okay, the part of the bone that is important, all right, to give it its structural rigidity. And of course, we need calcium to release our neurotransmitter, right, that's stored in the synaptic knobs. I can go on and on about calcium, but enough about calcium. Let's get back into the urinary system. All right, acid-base balance. We're going to talk about that quite a bit, a little bit in this chapter, but later on, we're really going to see, you know, when our pH level of our blood swings in either one direction, either to the acidic end or to the alkaline or basic end here, how the kidneys play a large role in that. So huge function for the kidneys. All right, regulation of blood pressure. Yeah, we talked about back in chapter 20, right, we were talking about regulation of blood pressure, the neurological component of it, and how the central nervous system plays a role in regulating blood pressure, but the kidneys play a really big role. So we'll talk about that. And then of course, elimination of biologically active molecules, any drugs that you might have to ingest, all right, and also hormones that you are producing here. Way back in chapter 7, we talked about, all right, vitamin D, the coveted vitamin D. And vitamin D has so many different properties and helpful qualities, one of which is the formation of calcitriol. That's the active form of vitamin D that helps, all right, with calcium reabsorption in the gut. All right, so the kidney plays a role in converting calcidiol into calcitriol. Erythropoietin, all right, that's again part of the endocrine function. Erythropoietin helps, all right, to increase the oxygen carrying capacity in our blood by generating and increasing our erythrocyte count, right, by producing more erythrocytes in the bone marrow. And then finally, this term here, gluconeogenesis, we're familiar with that because we talked about that when we've discussed different metabolic processes in the body. And gluconeogenesis primarily occurs in the liver. And up to this point, that's the only organ that I really mentioned about gluconeogenesis occurring in. But gluconeogenesis can, all right, when we're in a bad situation in which we are in a long-term fasting, or if you're in starvation mode, all right, then the kidneys will help, all right, the liver out in that process of gluconeogenesis. And basically what we're going to do is we are going to produce glucose from non-carbohydrate sources. It can be fatty acids, it can be proteins, amino acids, right? And so we talked about that before. All right, so let's talk about, all right, the actual filtering process here. And so we're going to discuss three terms here: filtrate, tubular fluid, and urine. All right, so it's important that you understand where, all right, these fluids are actually going to be generated, all right, and also where they're going. So with filtrate, excuse me, real simple with filtrate, that is the blood getting filtered, all right, primarily the plasma, okay, because the blood, as you know, is made up of several different things. You have the formed elements and then our blood plasma, all right? So pretty much we're going to filter out the blood plasma. And so as the blood flows through the glomerulus, and the glomerulus is going to be that capillary ball that has, that is kind of just wadded up, and then it's surrounded by a capsule, Bowman's capsule there. And so we're going to see how we're going to filter out that blood as it flows through that capillary ball, the glomerulus there. And primarily what we're going to see flowing out of the bloodstream is going to be water and then solutes, things that were dissolved into the blood plasma. And it's going to travel from the glomerulus into the glomerular space there, and then into the glomerular capsule there. And then as that occurs, that's what we call filtrate. All those substances, all that product that moves, all right, from the glomerulus, from the glomerular capillaries into that capsular space there. And then eventually we'll move it into the tubular system, right, of our kidneys, and we'll call it something else, right? Once it moves into that tubular system, all right, that first part of that tubular system that we have is the proximal convoluted tubule, also known as the PCT. Once it gets into there, easy to remember, all right, it's in the tubular system, so we call it tubular fluid. And so it's known as tubular fluid as it flows through, all right, the proximal convoluted tubule down into the nephron loop there, all right, where you're going to have both your the ascending and the descending branches there. And then we have our distal convoluted tubule, which will then drain into our collecting tubules. And then those collecting tubules will drain into the larger collecting ducts. And then as they are moving from the collecting ducts into the renal papilla there, around the minor calyces, then we call it urine. And pretty much once we form that urine, we are going to excrete that, right, out of the kidneys, right, down into the urinary bladder, and then out through the urethra there. So the fluid now, as it's moving through the papillary duct and entering into, all right, the renal sinus, primarily the minor calyx there, that's when we start to call it urine. And then the minor calyx will drain into the major calyx, and then several of the major calyces will drain into the renal pelvis. And then at the other end of the renal pelvis is our ureter. And that ureter will drain down into the urinary bladder. And you have a right and left ureter because you have a right and left kidney. And then pretty much the urinary bladder, its job is to store, all right, the urine until it's time for you to go to the bathroom. And then you'll excrete it out through the urethra there. So this slide here kind of shows you, all right, if we zoom in here, we'll see here's the glomerulus, all right, with Bowman's capsule surrounding it, or the glomerular capsule, and there's a space in between, all right, that wad of capillaries and the capsule. And that's the capsular space. And in that space, all right, that fluid is known as the filtrate. Once it works its way here into our tubular system here, then we call it, all right, the tubular fluid. And so all through here, here's the proximal convoluted tubule, drains down here into the nephron loop. And then we have our distal convoluted tubule. And then at the far end, we'll have our collecting tubules attached to our collecting duct. So when that fluid enters into any of those mentioned areas there, it's known as tubular fluid. Then we get down here to the renal papillae here and into our minor calyces. As it moves from that papillary duct into the minor calyce here, I can't zoom in anymore, around this region here, then that is when we refer to that fluid as urine. And then our minor calyces, several of those will drain into a larger structure called the major calyx. And then a couple of those major calyces will then all drain into the renal pelvis here. And then finally, that renal pelvis at the other end, all right, we'll have our ureter. and our urine will drain from the ureter down into the urinary bladder. And from the urinary bladder, once it's time to void the bladder, okay, it will move through the urethra here and then out of your body. All of that is known as urine. So now you have the kind of the big picture. But so now, of course, this course would not be this course unless we got down into the specifics, into the nitty-gritty. So now we're going to go through and discuss how we actually form this urine. And so there's three processes, and this is really important, and I really want you to understand, all right, these three terms here. All right, filtration, that's relatively easy. That's when we're just going to basically, all right, filter out the blood. All right, then we have resorption and then secretion. We'll get into those two terms. But basically, resorption is now that we have this fluid in our tubular system, we're going to reabsorb that fluid back into our bloodstream, not the major portion of our bloodstream, but we have these smaller blood vessels in the kidneys here. And then so the majority of the material that was filtered out will get reabsorbed into, all right, these blood vessels there. And then those blood vessels will be found in close proximity to our tubular system. And then along that way, some of the material might get secreted back into the tubular system. So these three terms, all right, you're going to have a general understanding. We'll work through them and I'll go over them with you, all right? But keep in mind, filtration, resorption, all right, and secretion. So let's start off, all right, at the beginning, way at the beginning, and we're going to talk about glomerular filtration. And this is going to happen in the glomerulus, right, where those capillaries are located. And so blood is flowing into the kidneys, okay, and it makes its way into the glomerular capillaries. And from there, right, we are going to filter out primarily water, right, and some of the stuff that was dissolved in the plasma of your blood. And so the water and those solutes will enter into that glomerular space. Then we change the name and call it filtrate. Now, keep in mind, all right, these substances and water are going to move from the capillaries into the space because of the difference in pressure from the vascular system there in the capillaries and that pressure found in that space there. The pressure will be higher in the capillaries than in that space. So that's going to promote filtration. That's going to promote water and the solutes moving from the bloodstream into that renal corpuscle or that capsular space found in the renal corpuscle so we can move them out of our bloodstream. Don't forget, we call that fluid filtrate. It's the only time we're going to call it filtrate, because from there, now we're going to move that material into our tube system. Right? So at that point, we refer to that substance as tubular fluid. And it'll be known as tubular fluid for a little while here. So during this part, or this portion of our filtration, I shouldn't say filtration process, but of the filtering process of the kidneys just in general, right? You're going to see three transport processes occurring: diffusion, osmosis, which are both passive, all right, transport processes. Right? Diffusion is involving solutes, osmosis is water. And then finally, we have active transport. So those three processes will be occurring in our tubular system during that tubular reabsorption period. So we're going to see things moving from the lumen of the tubules into, all right, back into our bloodstream there. So it's going to move from the lumen of the tubules, all right, across the walls of those structures, primarily between cells and through the cells, and it's going to return back to our blood. And the bloodstream structures that I referred to are called peritubular capillaries and vasa recta. The peritubular capillaries are obviously, look at the name, peritubular. They're going to be found around, right, primarily the tubular structures there that we just mentioned, you know, like the proximal convoluted tubule, the distal convoluted tubule. And the vasa recta is going to be primarily found around the nephron loop there. And so pretty much everything that you just filtered out will be reabsorbed. So most of the water will be, right? And anything that we consider, that your body considers to be vital, something important, right? Calcium, we just talked about calcium and all the roles that calcium plays. So your body considers it to be important. It's several other things, glucose, all right? Several different types of ions in addition to calcium, sodium, potassium. Right? So we're going to talk about all this. So all the stuff that your body needs and really wants, it's going to resorb. Makes sense. It wants to hold on to it. You're going to need it for stuff. But the stuff that you don't need and any metabolic waste that you might have had floating around in your blood, okay, for example, urea, right? You don't need that. So some of that stuff is going to remain in the lumen there of the tubular system. It's going to remain as part of the tubular fluid. So excess solutes, waste products, and some of the water. Again, if your body has excess sodium and it doesn't need anymore or want anymore, it's going to excrete it. It's going to get rid of it. But if it's low on sodium, it's going to resorb, all right, as much sodium as it possibly can, all right, so it won't get rid of that. All right, then finally, the third part of our urine formation is going to be secretion, tubular secretion. And this is when we are going to move stuff that was reabsorbed into the bloodstream. We're going to then secrete it back into, all right, the tubular lumen there. So this process is mainly governed by active transport. And active transport, as you know, involves the use of ATP. So we're going to move stuff from the blood, from the vasa recta, from the peritubular capillaries, and we're going to move it from those blood vessels back into our tubular system. So it's basically, we're going to return some of that stuff to the tubular fluid. And then that material will remain in the tubular fluid, and then eventually it will move into the collecting ducts and then eventually become part of our urine. Keep in mind, this is a selective process. We're going to get into all this fun stuff. This is just, again, a nice overview as to what we're going to see. All right, you're going to be really amazed about how important the urinary system is. Your kidneys are fascinating organs. They are just so impressive. So this is a great picture. I love this picture because it kind of shows you, all right, all those three processes: glomerular filtration, tubular resorption, and then tubular secretion. And so you can see here, here is our glomerulus. You have your afferent arterial, and that's bringing the blood, all right, into the glomerulus here. Notice the diameter of the afferent arterial is larger than the diameter of the efferent arterial. Easy way to remember the name of this blood vessel is efferent. It's exiting the glomerulus. All right, so you'll have, all right, more blood flowing into the glomerulus because of that wider lumen there. So this is showing us as the blood enters into the lumen, okay, we're going to then filter out the blood plasma, and water and solutes will move across into the space here, all right, the glomerular space or Bowman space, and then that's when it's known as filtrate. And then as it moves into our tubular system here, that's when we call it tubular fluid. And so you'll notice the purple arrows here are indicating tubular resorption. So a lot of material that was filtered out is going to move back into, all right, your nephron or your kidney vascular system. Move right back into these blood vessels, these peritubular capillaries, or in the vasa recta capillaries down here by the nephron loop. And so you'll see that there's pretty much a decent amount of movement in several areas of water and those solutes going back into the blood. Then you'll notice also these tan-looking arrows are representing tubular secretion, where we are going to be moving material from the blood vessels, the peritubular capillaries and the vasa recta, into our tubular system here. And so we're going to talk about when this occurs, where this occurs, what causes all this, right? But just keep in mind, you are going to see movement of materials back and forth. And a lot of that is going to be determined on what the body needs, what's going on. And we'll talk about those situations. All right, so let's start way at the beginning with the first process of filtration. So in order to better understand filtration, we need to talk about our filtration membrane. And so that's going to be like our sieve. That's going to, determine what is allowed to leave the blood and what has to stay in the blood. So a couple of the characteristics that we need to see for our filtration membrane is porous is one. Well, that makes sense, right? Because we want things to be able to move across. All right, thin always makes sense. Keep in mind, we learned about that concept in chapter 5 when we talked about epithelium. Simple epithelium is way better at filtering than stratified epithelium because it's a thin barrier. And then negatively charged structure. Now this is interesting, and that primarily has to do with proteins. Proteins are normally negatively charged. And so as you know, like charges repel each other. So we really don't want to be filtering out proteins from the blood. And if you are finding protein in your urine, there's usually something going on that shouldn't be going on. But so those are the characteristics that we're looking at. Porous, thin, and negatively charged structure for our filtration membrane. And so our filtration membrane is going to be made up of the glomerulus, all right, those are the blood vessels, and then the visceral layer of our glomerular capsule. And of course, there's going to be, it's not really part of the membrane, but we'll talk about this, right, in a little bit, but we have our glomerular space, right? We'll get to that. All right, so let's look at the layers of the filtration membrane, starting with the inside layer. Endothelium. What is endothelium? Endothelium is epithelium that lines vessel walls. So since we're talking about capillaries, we have to talk about, all right, the epithelial tissue that is found in the glomerulus, right? Well, that is going to be made up of, right, a capillary is going to be simple squamous epithelium. So this type of tissue, this endothelium, is going to be fenestrated. We talked about fenestrated capillaries a while back in our circulatory system chapter when we were describing the 3 different types of capillaries. Fenestrated means we have a capillary that has these holes in it. Again, we want to have holes. This is a good time to have a leaky capillary. Right, so our capillary has these holes, these fenestrations that allow, all right, certain materials to leak out of the bloodstream. Pretty much what it's going to allow is stuff from the plasma, primarily water and anything that was dissolved in the plasma, right? And some of the really small substances to pass through. We are going to prevent the movement of really large items, right? Our formed elements of our blood, white blood cells, red blood cells, right? Even platelets, we are going to restrict the movement of those large structures across this layer of our filtration membrane here. All right, so as you recall, epithelium always sits on a basement membrane. So the endothelium here of the glomerulus sits on a basement membrane made up of glycoproteins and proteoglycan molecules. And so these structures are going to prevent, again, all right, the movement, all right, of large proteins. We don't want proteins leaking out of our blood because that can affect our the oncotic colloid pressure there. And then that can cause a whole host of issues occurring. So the basement membrane prevents the movement of large plasma proteins, right, from the lumen of the capillaries into that glomerular space. And then finally, we have our visceral layer, right? And the visceral layer, right, wraps around those capillaries. And we have these cells there called podocytes. And these podocytes have these appendages, all right, called pedicels. And so basically what they do is they allow for these slit-like structures. We call those filtration slits. And so they create these filtration slits, these structures here, right, to allow, all right, for the passage of certain materials, but not others. But the nice thing is they help to restrict small proteins from moving across the glomerular space there. And then it's worth mentioning, all right, the mesengial cells. These cells, right, are going to be found all throughout those little loop structures there in the glomerulus. And their job, there's several jobs that they have, all right. One is phagocytic. So if stuff gets stuck in there, right, they'll go and gobble it up, right. Contractile, and we'll talk about that here in a little bit when we talk about the glomerular filtration, all right, rates, and then also some signaling properties. And again, we'll talk about those properties in a little bit, right? So the mesengial cells play a large role, but they are found, all right, in the nooks and crannies, those capillary loops there. And their job is to gobble up stuff that gets stuck and doesn't make it into the filtrate. All right, we've got to keep our filtering system nice and clear so they'll eat things up. And then they have this contractile property that's going to help with our glomerular filtration rate, a concept that we'll talk about later on here. So here's a great picture of our filtration system here. And we'll see here, right, here is the capillary, okay? And we have, and you notice I called it fenestrated, so it has these holes in it. And so that will allow, all right, for the blood plasma and dissolved substances to move from the lumen of the capillary outwards. And then, of course, that endothelium there sits on the basement membrane, and that basement membrane is going to help to restrict the movement of large proteins. And then you'll see the podocyte sitting on the outermost area of our filtration membrane. And here are those pedicels. You can see these nice extensions that come off of these processes here, and they create these filtration slits. And so these filtration slits, all right, prevent the movement of those small proteins. Now let's zoom in a little bit. I want to show you a nice cross section of this. I love this picture. All right, so you can see here, all right, when we're looking, here's the inside of our capillary. You can see, look at these large foreign blood elements. All right, leukocyte, red blood cell, platelets. They can't fit through, all right, these fenestrations here by design, So what can make it through is going to be, all right, obviously water, right, glucose molecules, all right, a lot of our water-soluble vitamins will make it through, all right, even some small proteins. We don't want, I mean, they can be pretty small, they can slide through, but the large proteins pretty much blocked. And if they're large enough to get through the fenestrations, that basement membrane is going to prevent the movement of those large proteins from going through. And then they have to make it through one more part, and those are going to be the filtration slits here of the podocyte that was created by the podocyte. And so the small proteins will pretty much be prevented from making it through there. So here, what we want to see are these structures here: water, glucose, amino acid, ions, urea, that's metabolic waste here. Some of the hormones are water-soluble vitamins, B and C, all right, ketones. You'll get small amounts of small proteins, but the larger proteins should be prevented from making it through. And all of that will make it here into the capsular space there. So it's a pretty interesting system, pretty effective also. So this is a pretty decent number here. 180 liters of filtrate are produced every day. Every day, your kidneys are working to make this happen. Right, so most of this material, right, are going to be solutes, and we'll even see, like I just mentioned, water-soluble vitamins, but very little. So this is important, minimal amounts of protein. You do a urinalysis, if you've ever had a urinalysis done in which they check what what's in your urine. Is there blood in your urine? Is there glucose in your urine? One of the things they look for is protein. And normally you should not have protein in your urine. You should have minimal amounts, very, very little. Right, so the filtrate moves through the capsular space, and it's going to be funneled into the proximal convoluted tubule. So what we're going to see then, anything that wasn't filtered out stays in the glomerulus, moves through that network of capillaries there, and then it eventually exits out of the renal corpuscle through our exiting blood vessel called the efferent arteriole. Efferent, exits. Afferent is entering into the glomerulus. And like I said, any of that material that gets filtered out somewhat or gets trapped, all right, the mesengial cells come through and gobble it up. So when we're looking at our blood, all right, the substances that are in our blood, we have three categories for them: freely filtered, not filtered, and limited filtration. So freely filtered, and again, how I kind of remember this, or the concept that was similar, we talked about material that can move across the plasma membrane, that can diffuse directly through the plasma membrane, has to be small and nonpolar, all right? So similarly here, small substances are freely filtered. They can just move right through, right, without being hindered. Water, obviously, okay, glucose, amino acids, ions, vitamin B, vitamin C, right, some of the urea, they can make it right through, and again, by design. Things that cannot be filtered, obviously, are going to be your formed blood elements, the erythrocytes, your white blood cells, your leukocytes, your platelets, right, large proteins. They cannot make it through that filtration membrane, right? Some of the things that are going to be limited, right, but may make it through, are going to be proteins. It depends on the size then. Obviously, the large proteins, no bueno, right? But some of the smaller, real small proteins can make it through. Now, a lot of the times proteins are prevented from making it through mainly because they're big. But also, I mentioned this before, going back to some of the characteristics that we want to see in our filtration membrane. One was it's negatively charged. Well, guess what? Proteins are also negatively charged, and so they'll repel each other. And so that helps to prevent the movement, right, of those proteins through that filtration membrane. So it does come in handy to have a negative charge there. HP with a small subscript letter. I remember bulk flow, right? Well, guess what? We can't get away from, right, pressure gradients in this chapter because we're going to talk about it. So now we're going to talk about, right, the concept of glomerular filtration and how pressures, right, are going to affect glomerular filtration. So we're going to discuss glomerular hydrostatic, all right, blood pressure, which is known as HPG, small g. So this is going to be the pressure of the blood in the glomerulus there, in those capillaries. And hydrostatic pressure pushes things against the wall of the container of whatever it's inside, like the pipes in your house. The hydrostatic pressure of water is going to be the pressure that water is pushing on the pipes in your house. So what we're going to see here, all right, in the glomerulus, we're talking about the glomerular hydrostatic pressure. We're going to be talking about that pressure that the water and the solutes, the blood plasma, is going to be pressing on the walls there of the glomerulus there. And basically, like I said, you want to see a larger pressure in the glomerulus than in the capsular space there to promote filtration. All right, so as long as that pressure is higher in the glomerulus and it's less in the capsular space, then we can move, all right, materials from the blood into the capsular space. All right, so again, we have to have that higher pressure in our glomerulus. Now, this is interesting, right, because we talked about bulk flow and how pressures work when we were discussing the circulatory system and our capillary beds and where the tissues reside. We had that systemic capillary bed that the pressure was relatively high, all right, at the arterial end to move, all right, substances out of the blood and into the tissues. But at the other end of our capillary bed, right, the pressure would be lower, so we could move material from, all right, the tissues into the capillary bed and then into our venous circulatory system here. All right, but what we're going to see here, it's going to be always that the pressure of blood in our glomerulus is going to be higher than what we would see in our systemic capillary beds. And again, we need that to occur so we can get filtration, right? And again, another thing, and I brought this to your attention before when I was talking about the afferent arterial and the efferent arterial. The afferent arterial has a larger diameter. The larger the diameter of a blood vessel, the greater the amount of blood flow in that area, so you'll have higher pressure, right? The smaller the diameter, right, you'll have less flow. You'll have greater resistance, right, but you'll have less flow. All right, so as we're talking, some other terms that you're familiar with, all right, blood colloid osmotic pressure, right, osmotic pressure. Anytime you hear osmotic pressure, always think of the movement of water, right? And two things will affect water's movement. Quick review, chapter 4. All right, osmotic pressure is going to be when water moves from an area of high concentration to low concentration. That's going to cause that flow of water occurring there, but also you have to take into consideration that water is always going to move to the area where there's a higher solute concentration. And that's what we're seeing here. Our osmotic pressure, right, is going to be governed by those dissolved solutes in that fluid there. And so the dissolved solutes are going to influence water's movement. So what we'll see here, all right, that there's going to be quite a bit of dissolved solutes in your blood. So water, right, is not necessarily going to freely flow from the glomerulus into the capsular space there. We'll see some opposition to that filtration because water, remember, water is a follower. Water is a follower. It likes to follow sodium. It likes to follow glucose. And so, of course, all right, it's going to, if some of those materials are being filtered out, you're going to get water that will follow that. But if we're still retaining, all right, those materials in your blood, all right, water will also follow that. Okay, it can't make up its mind, so it just follows whatever. All right, so this, we say that the blood colloid osmotic pressure is going to draw back some of that fluid into the glomerulus. So we're going to be opposing filtration. And then we have our capsular hydrostatic pressure. Now, this is the pressure that we're going to see, all right, out in the glomerular capsule there. And that's because whatever was filtered out, right, that filtrate there, as it is accumulating in that area, we'll start to see it impeding the movement of any fluid, right, because that increased pressure out there in the glomerular capsule, right, will start to oppose further movement of more fluid coming out of the glomerulus here. I got a picture here that I'll show you here in a second, give you a better idea. All right, so here's our equation, right, for our net filtration pressure. Obviously, if it's a positive number, we will be promoting filtration. We always want to promote filtration here, right? So our NFP, the net filtration pressure, here's our hydrostatic pressure out there in the glomerulus, okay? And then we're going to take the difference of the sums of the oncotic, osmotic pressure, or excuse me, the osmotic colloid pressure in the glomerulus. And we're also going to take the hydrostatic pressure that's out in the capsular space. So we're going to add up those two numbers and then subtract that from the hydrostatic pressure out there in the glomerulus there. That is correct. Yeah, that is, yes, Kristen. The negative, we would not see, all right, filtration, but it would be resorption. You nailed it. You go. So here we can see our value, positive 10 millimeters of mercury, and that's telling us that this is favoring filtration. If this number was not 50, but if it was 70, all right, then we would have a negative 10 value, and then that would not be favoring filtration. That would be opposing filtration. So this picture here again is showing you. Zoom in a little bit. Okay, here's the hydrostatic pressure of the glomerulus. All right, all that fluid pushing to get out of the capillaries there and into this space here. So you'll see we take the value, 60 millimeters of mercury, and then we calculate here is the osmotic colloid pressure, everything that's still in the colloid, excuse me, in the capillaries here. You know, all those proteins and solutes that are trying to pull fluid back in, primarily water. We calculate that out. And then as fluid has been moving out into the glomerular space here, right, it's accumulating here, and that fluid's presence here starts to increase the pressure out here. And so that is going to be our hydrostatic pressure of the capsular space here, or the glomerular capsule. And so that also will oppose the hydrostatic pressure. But as long as we have a positive value here, we'll have filtration. And so we'll see, because we have that filtration value, then our fluid will move out. All right, that brings me into the glomerular filtration rate. Glomerular filtration rate. So we're now going to discuss, we're going to stay with pressures and whatnot, but this concept is very important. So I'm going to kind of move through this nice and easy. All right, so that equation that we just talked about right here, the net filtration pressure, all right, that's basically the pressure of all this filtering process going on in the glomerulus, moving fluids and substances out of the bloodstream and into the tubular system. Now we're going to talk about what can affect some of the variables that can affect that net filtration pressure. So to do so, we have to talk about our glomerular filtration rate. And whenever we mention this word rate, time is involved. And so this is going to be the amount or the volume of filtrate that is going to be generated or created, all right, over a unit of time, and that's usually going to be 1 minute. So every minute we're going to make X amount of filtrate. All right, so some of the things, all right, that will increase our net filtration pressure. One is if we increase our glomerular filtration rate. We'll talk about how we do that. All right. Also, right, if we increase, a lot of this involves increasing stuff, right? If we increase, all right, our solutes in our fluid there and the water, okay, that's going to be found in the tubular fluid, right? If we increase solutes in the water in the tubular fluid, all right, if we increase substances that are found in the urine, and then finally, if we decrease resorption. So what does that mean? If we decrease, all right, the filtrate that is getting resorbed from the tubular system back into the blood. Okay, so these four things: increase in GFR, increase the solutes in water remaining in the tubular fluid, if we increase the substances in our urine, and if we decrease the filtration resorption. All right, so let's talk about glomerular filtration rate. And this is very important. And this term at the top here, all right, or the sentence at the top, I really want you to kind of hold on to that. And I want to explain to you why this is important. But glomerular filtration rate is tightly regulated. We always want to keep it at a certain rate. All right, so we're going to talk about what happens when certain things are occurring. All right, it's like that guy that's trying to balance something and juggling. You keep adding stuff to whatever it is that he's trying to keep balanced, all right, or do something to his body that he's trying to keep balanced. As long as he keeps maintaining what's going on, everything's okay. So this is what we're going to see. We're going to determine, all right, how our kidneys, all right, are going to be filtering out your blood, but we keep it in a very regulated format here. All right, and it's based on the needs of the body, whatever's going on. And you might notice there's certain times where you're like, hey, I'm peeing a lot, right? And that might be because you're drinking a lot of water. Or you might notice, hey, I'm not really peeing that much. Well, gosh darn it, I haven't been drinking any fluids today. So this is kind of like what we're going to talk about. We're going to talk about how, all right, your kidneys are going to be producing in the urine depending on what is occurring in your body physiologically, how much water you're taking in versus maybe how little water you're taking in. All right. So when we talk about our glomerular filtration rate, a couple things are going to affect it. Right. One is going to be how big or small the afferent arteriole is. So we can affect the glomerular filtration rate depending on how much vasoconstriction occurs in the afferent arteriole or how much vasodilation occurs in the afferent arteriole. And in case you're forgetting, what is the afferent arteriole? That's this fellow right here. That's the blood vessel that's bringing blood into our filtration mechanism, into our glomerulus. So we're going to affect, all right, how big or how small that is. And in doing so, that will affect our glomerular filtration rate. Also, we can also influence our glomerular filtration rate by effectively altering the surface area of the filtration membrane. All right, and so this is where some of those mesengial cells are going to come into play here. All right, we're going to talk about that. All right, so there's two ways that we are going to look at the kidney on how it's able to regulate itself. We have intrinsic controls, and that means everything that's going on inside the kidney. And then we have our extrinsic controls, and this is happening outside the kidney. So we'll start off with what's going on inside the kidney. And so in determining our glomerular filtration rate, how do we want to regulate it? And again, we always want to try to keep it, just like our blood pressure. We always want to keep our blood pressure within a certain range, right? You always want to keep your body temperature within a certain range, right? We want to also keep our glomerular filtration rate within a certain range. We want to make it constant and consistent. So our kidneys, all right, through the intrinsic controls, are going to display what we call renal autoregulation. Renal autoregulation. And so this is how your kidney is going to be able to maintain two things: constant blood pressure and constant glomerular filtration rate. Like I said, we always want to maintain, right, the glomerular filtration rate at a consistent, constant level. So you could have all these changes occurring outside the kidneys. Your blood pressure could be going up, it could be going down, but we're always going to try to maintain the blood pressure inside your kidneys and the glomerular filtration rate inside your kidneys at a constant level. So we have two mechanisms that help to do this, right? The myogenic response and then our tubular glomerular feedback mechanism. Now, myogenic, think MYO, myo is muscle. So we're going to involve some muscle. Well, it's not going to be skeletal muscle and it's not going to be cardiac muscle because we're not in the heart. It's going to be smooth muscle. And so the only structure that we're familiar with that has smooth muscle in it is going to be our circulatory system within the kidney. And we've only really talked about a few. So we're going to deal with the afferent arteriole. So we're going to either contract or relax that smooth muscle in the afferent arteriole. And it's doing this in response to the amount of stretch that is occurring in the blood vessel wall. All right, how much stretch is occurring? So if we're increasing the stretch or decreasing the stretch. So we're going to start off, all right, by talking about what happens when we decrease the blood pressure in the afferent arteriole. If we decrease the pressure, we decrease the stretch. So here's what we're going to see. We decrease that stretch, our smooth muscle cells within the afferent arteriole, all right, they relax, and that blood vessel undergoes vasodilation. A larger lumen, what does that mean? Well, that means more blood can flow into that blood vessel. So that's what we see. When the afferent arteriole vasodilates, it allows more blood to enter into the glomerulus. And so this compensates for that drop or that decrease in the blood pressure. Because remember, this is our goal, folks. We always want to maintain a constant glomerular filtration rate. We always want to maintain that. So when our blood pressure decreases, right, we are going to then see vasodilation occur that allows more blood to flow into the glomerulus. So that decrease in blood pressure, all right, that's occurring, it allows the glomerular filtration rate to remain normal. Pretty cool, huh? So let's talk about what happens when we increase our blood pressure. All right, so now you're exercising, let's say, and your blood pressure starts to increase. Okay, so we get an increase in blood pressure. So we're going to see, right, increase in pressure means more stretch on the blood vessel wall. And because of that increase in stretch, now we're going to see the smooth muscle cells contract. So we're going to get vasoconstriction. Vasoconstriction now is going to result in less blood getting into the glomerulus. Even though your systemic blood pressure has increased, right, your kidneys are not going to let that affect the filtration rates. And so they're going to cause vasoconstriction, all right? So less blood makes it into the glomerulus. So again, ultimately, this is what we're always trying to do. We're always trying to maintain that normal range of our glomerular filtration rate. So let's look here, right in the middle. This is where we're going to start off with first, right here. Normal systemic blood pressure. Everything's good to go. We're not worried about everything or anything, I should say. All right, so blood enters in through the afferent arteriole. Your blood pressure is at 120/80. It circulates through, right? We're getting our normal glomerular filtration rate occurring. Things are getting filtered out at the normal rate. We're happy with that, right? So now what happens is your systemic blood pressure has decreased. Systemic blood pressure is decreased to, let's say, 90/60. Right? It's quite a drop. So because there's less systemic blood pressure, right, that decrease in pressure causes the afferent arteriole to vasodilate. Look at the size here, right? That's nice and medium size. Look how big this is. So that opens up, allows more blood to flow through, because we always want to keep that glomerular filtration rate at a consistent level, at its normal level. So we open this up, allows more blood to flow through, right? And it keeps the glomerular filtration rate going at its normal rates. Now, here we see a situation where you're out there exercising, you're under a lot of stress, let's say, for whatever reason, or you're out there for a run, working out, whatever. Your systemic blood pressure goes up to 148/92. So blood pressure increases, right, puts more pressure on the vessel wall, right? So what happens in response to that is the afferent arteriole vasoconstricts. Again, you can look at the normal, right, pretty decent lumen size, but now it vasoconstricts, it closes down, lets less blood in. Again, keeping in mind that we always want to keep our glomerular filtration rate at a constant level. So this compensates by causing vasoconstriction, decreases blood flow into this area here, all right, and you can see we'll maintain that normal glomerular filtration rate. So that's the myogenic response. The other response that we can see is called the tubuloglomerular feedback mechanism. This is our backup to the backup, okay? So this system, all right, we're going to involve, all right, our macula densa cells here. So let's go with another example, all right? So blood pressure now is increased, and we'll see because of that blood pressure increasing, all right, sodium chloride, essentially salt, right? We're going to see a higher amount of that sodium chloride in the tubular fluid. And because of that, all right, these macula densa cells, they monitor, all right, the chemical makeup of that tubular fluid here. And so what they'll do then is they've noticed, hey, you know what, there's a larger amount, all right, of sodium chloride, of salt, in the tubular fluid, a little bit more than normal, right? Something's not quite right here, no problem. It will then cause vasoconstriction in the efferent arteriole, right, to try to close that off so we can decrease, right, the glomerular filtration rate. Or I should say decrease it, but to keep it constant. All right. Let's check out the regulation of the glomerular filtration rate here when we're talking about, hold on one second, folks. Okay, so what are we seeing when we're looking at the glomerular filtration rate? There are some limitations to it, you know, and so when we're dealing with autoregulation, right, again, we want to, like I said, we want to maintain that glomerular filtration rate within a certain range here. So when we're talking about our autoregulation, right, this is the range that of the pressure that we want to see, right, within, all right, the actual glomerulus. Excuse me, I can't talk today. So when that pressure drops below 80 millimeters, unfortunately, we run into a problem. We can only get to a certain amount of dilation. You can't make your blood vessels dilate, you know, infinitely. So when the blood pressure gets very low, all right, at 80 millimeters of mercury, we've reached maximum dilation here. And so what will happen is, right, at that point, we'll see pretty much in the urine, pretty much nothing, all right, in the urine there, because we've maximized everything in regards to the blood vessel dilation. And so, of course, right, glomerular pressure, I'll show you a little graph here in a moment, decreases. And so as a result, if the glomerular blood pressure decreases, our glomerular filtration rate will also decrease because that causes that hydrostatic pressure, right, to have dropped down. So what we'll see conversely, when we increase our blood pressure above 180, the opposite occurs. We get maximum constriction, right? And so what we'll see in that situation is we've constricted as far as we can, right? And of course, what we'll see is that increase in glomerular blood pressure and glomerular filtration rate, you're going to be pushing out so much urine, all right, it'll make your head spin. Again, a lot of it has to do with fluid intake. There's some pathological conditions that cause that, but this is what we're seeing with our autoregulation. Ideally, okay, when we're having our normal urine production here, okay, autoregulation is chugging along great. But, right, when our pressure just drops way off, okay, here, you will see symptomatically wise that you're hardly producing any urine and you're not getting really rid of any waste. If you are producing any urine, it's going to be in a very, very small amount. And then, of course, on the other end, all right, when we get maximum vasoconstriction going on, above 180 millimeters of mercury in the pressure there, okay, you will be excreting all sorts of urine. And so your urine rate will, or volume, will increase quite a bit. All right, so we talked about the intrinsic controls. Let's get into the extrinsic controls. Let's talk about our control centers, both the nervous system and our endocrine system. How do they get involved? All right, how are we going to, again, with the intrinsic controls, we're trying to keep the glomerular filtration rate consistent, and we're going to maintain that. This is what we're trying to do. We're trying to maintain this right here. We're trying to maintain the blue. That's what we're looking at. This value. We want to keep it right in that area there. All right, so that's maintain. Autoregulation is maintain. All right, but now when we get to the extrinsic, all right, controls, now, all right, we have to deal with what happens when our glomerular filtration rate changes. All right, so now we're going to be changing some stuff. So let's talk about our nervous system, all right, specifically the autonomic nervous system, and how, all right, we are going to see a decreasing GFR due to our fight or flight division, the sympathetic nervous system, when it stimulates. So I like to use exercise, okay, when you're working out. And as you are stimulating the sympathetic nervous system through being chased by a bear or through going for a run, okay, we're going to see a decrease in our GFR. And this is a result of vasoconstriction. Remember, the sympathetic nervous system is the only part of the nervous autonomic nervous system that can affect the lumen diameter of your blood vessels. So we're going to see vasoconstriction of both the afferent and, in this time, the efferent arterioles here. Right? So there is a group of cells called the granular cells, and they're in the juxtaglomerular apparatus here. And so those are the ones that are going to produce this fella right here, renin. Remember renin? Renin is going to convert, all right, angiotensinogen into angiotensin 1, and then angiotensin 1 is going to circulate into the lungs, get converted by the ACE enzyme there, and it converts angiotensin 1 into angiotensin 2. And so angiotensin 2 makes its way back to the kidneys, and it's going to cause the mesengial cells to contract. Remember those qualities or characteristics of the mesengial cells? They're phagocytic, right? They are contractile, and they have chemical signaling properties. Well, this is the contractile characteristic. So they are going to contract, and what that does is that decreases our surface area of the glomerulus. And when we decrease the surface area of the glomerulus, we decrease our glomerular filtration rate. And so this is why you have to pee less when you're running. Not the only reason, all right? But again, all right, your body is going to conserve water, which makes sense, all right? Especially if you're working out and running, you don't want to become severely dehydrated. So how do we increase our glomerular filtration rate? Well, you remember this wonderful little ditty here, the atrial natriuretic peptide. That's that hormone that's produced right, by the cardiac muscle cells and the walls of the atria when there is more blood volume, and increased stretch on the atrial walls, right? So what we'll see here is that the ANP is going to cause relaxation of the afferent arteriole, that vasodilation that occurs, all right? What we'll see is an inhibition of renin being released. So now we're not making angiotensin II, okay? And so that causes the mesengial cells to undergo relaxation that will increase our surface area for the filtration membrane. And if we increase that surface area, we're going to increase our GFR. And so this now will then result in increased urine volume. And by doing so, if you're excreting more fluid, you're going to decrease your blood volume, and you will therefore decrease your blood pressure. So here you can see how we're going to decrease, all right, GFR from our sympathetic stimulation here, okay? We're working out, running, all right? And so we'll see how that working out and that running there, all right, is going to cause the sympathetic nervous system to cause vasoconstriction in both the efferent, or excuse me, the afferent and also the efferent. I know it's not indicated here. So we'll see decrease in blood flow here. And so that will also then cause the granular cells there and the juxtaglomerular apparatus there, right, to release the renin, and that renin is going to then cause the production of angiotensin II to occur, and that angiotensin II is going to cause the mesengial cells to contract, therefore decreasing, right, the surface area there. And decreasing that surface area, then we're going to decrease the glomerular filtration rate, and therefore we'll have less urine being produced. And therefore, because of that decrease in urine production, right, we are going to see, because we have a decrease in the filtration surface area there, we're going to retain more blood volume because we're not getting rid of that fluid there into the filtrate. Will increase our blood pressure. All right, and then again, you can see when we're increasing the GFR, all right, through ANP, right, that ANP is being produced because of the stretch on the arterial wall, and we will then see vasodilation of the efferent arteriole. More blood is able to make it into the glomerulus, right? That it's going to inhibit renin from being produced. And so we'll see a decrease in angiotensin II that causes relaxation of the mesengial cells. And so the relaxation of the mesengial cells will cause that increase in the surface area filtration. And as a result of that, your fluid, all right, the filtrate will increase, which means your urine production will increase, which will mean that your blood volume will decrease, and therefore the blood pressure will decrease. Right, so a lot of stuff there going on with, all right, blood pressure, glomerular filtration rate. I want to move on here just real quick. I'm going to be wrapping it up here in the next few minutes, but I want to move on to our what we call the transport process. We're now going to talk about what is going to influence, all right, that tubular reabsorption and the tubular secretion. Remember, resorption is when material is moving from the tubular system into the blood, and secretion is when that material, those solutes, those substances are moving from the blood back into our tubular system here. So when we're looking at the tubular system here, all right, it is made up of simple epithelium. Hooray, simple. We know simple epithelium is great for filtration, secretion, and absorption. Right, so this is going to be the ideal type of tissue, right, that's going to be found along our tubular wall here. It's going to be that simple epithelium. So we have, all right, two ways that the material, okay, or the substances can move from the tubular system into the blood, and vice versa. We have what's known as paracellular, and so this is going to move things between the cells. And then transcellular, is going to move substances across the epithelial cells. And so we have to address, right, when we're looking at our transcellular transport, these cells that we're looking at are going to be cuboidal in shape. So the edge of the epithelium that's in the lumen is going to be referred to as the luminal membrane. It obviously is going to contact the tubular fluid. And then the other part of it, what we call the basolateral membrane, right? That is going to contact the basement membrane. And then on the other side of the basement membrane will be your capillaries, your blood. So depending on which way we're going, if we're undergoing resorption, we're going to move from the lumen into the blood. So we will see things go from the luminal membrane into the cell and then across the basolateral membrane. Now, if we're talking about secretion, then we're just going to flip it. We're going to go from the blood across the basolateral membrane and then into, all right, the lumen across the luminal membrane. And so we'll talk about that, okay? So this is important because we're going to discuss here, right, shortly, how certain substances are going to move throughout these structures here. Because again, your body's going to want to keep certain things and it's going to want to get rid of certain things. And so in these cells, right, we will see our transport proteins. What are those? Channels, okay? Channels are going, remember, transport proteins, all right, proteins have several functions. One of those functions is movement. And when we're talking about movement, one of the ideals for movement is going to be transport proteins. So transport proteins can be pumps, they can be channels. Right? So these things we're going to talk now in a little bit more detail. All right, so we're going to be dealing with transport proteins that are going to be located either on the luminal or the basolateral membranes, in some cases both. Then we have our peritubular capillaries, and those are going to be capillaries, all right, that are going to be found, all right, around our tubular system here. And so we're going to be dealing with, this is important that you understand this. The peritubular capillaries are going to have a low hydrostatic pressure and a high oncotic pressure. What does that mean? That, well, if you have a high oncotic pressure, that means you have a lot of stuff inside of that capillary. All right, the low hydrostatic pressure means, all right, it's harder for, there's less fluid, all right, in the capillaries pushing outwards. So in this situation, these peritubular capillaries are going to promote or facilitate resorption through, remember, bulk flow. You know what bulk flow is. All right, so this is important to know this concept. Most resorption is going to occur in the proximal convoluted tubule. And to make it even better, we're going to increase our surface area by throwing a bunch of microvilli onto our cuboidal epithelium there. So instead of our surface area for our cell being flat, all right, now what we'll do is we're just going to make our surface area increase with these microvilli here. And very, very clever. All right, so this is what we're going to see in this picture here. I love this picture because it shows you, all right, the pericellular transport, stuff moving in between the cells, going back and forth. Then we have our transcellular transport. You can see how we have our transport proteins, channels or pumps, right, that are going to be moving things. This is the luminal surface here, because here's our lumen, the luminal membrane, and then our basolateral membrane, all right, is going to be here at the bottom. Okay, and that's going to make contact with the basement membrane. And then on the other side of the basement membrane is our blood vessel here. So you can see here, all right, our purple area here is showing us, right, tubular resorption, things moving from the lumen of the tubule back into our peritubular capillaries, into the blood. And then tubular secretion is going to be moving stuff from the blood back into the lumen here. And so we're going to talk about all that affects if it's going to be undergoing tubular resorption or tubular secretion. Right? Keep in mind, when we're dealing with the peritubular capillary, we have a low hydrostatic pressure. So the force of fluid on the walls here of the capillary is relatively low, but it has a high colloid osmotic pressure. So that's the pulling force, trying to suck things into the capillary. All right, so we're going to see that is a high pressure. All right, so we have these two terms here, okay? Transport maximum, okay, known as T subscript M, and then our renal threshold. Now, the transport maximum, this is important. This is the maximum rates that a substance can be resorbed or secreted, right, over obviously a certain period of time. So we see a lot of this with glucose, for example. All right. Now, again, when we're dealing with transport maximum, we can only, there is a limit to what we can do when it comes to resorbing material into the bloodstream or secreting it from the bloodstream back into the tubular system there. And so the transport maximum, all right, is based on the number of transport proteins that you can stick in the cell membrane of those cells that line our tubular system here. So for example, all right, it says here, if no more than 375 milligrams per minute, all right, of glucose can be reabsorbed, meaning, all right, we put in as many transport proteins into the plasma membrane as possible, but yet, all right, there is still not enough transport proteins, okay, in the plasma membrane to resorb that glucose from the tubular fluid. So if there's still too much, right, if it is greater than 375 milligrams per minute, then we are going to see glucose show up in the urine. That's what we see with diabetics, right? Their blood sugar levels are so high, and the kidney tries to resorb as much glucose because it wants to. It doesn't want to excrete glucose. It wants to keep the glucose. Okay, but there's just too much in your blood, and those cells have put in the maximum number of transport proteins into their plasma membrane. They can't fit any more in there, right? So it can't stop the glucose from being excreted in the urine. So if you don't have any glucose in your urine, then you have to assume that the cells have a large enough number of transport proteins in which it's able to resorb all the glucose from the tubular fluid. So that's what we're talking about here with renal threshold. All right, renal threshold basically is telling us, all right, the maximum amount of whatever it is that we're monitoring in our plasma, in our blood plasma, okay, that is going to be transported into the blood from the tubule lumen there, all right. This value is going to be how much that of whatever it is that we're monitoring, I guess you could say, without seeing any of that in the urine. So that's our renal threshold, meaning, just like I said, if you are able to resorb everything, okay, then we won't see it in the urine. That means we haven't exceeded our transport maximum. So that brings me to glucosuria. All right, again, glucosuria, obviously, urea has to do with our urine, and gluco, right, is the amount of glucose. So what we're looking at here, all right, when our blood glucose levels exceed 300 milligrams per deciliter, all right, so 350, 400, all right, above that value of 300 milligrams per deciliter, we will start to see, all right, glucose in our urine. If we're below the value of 300 millimeters, if we're at 200, right, we should not see any glucose in the urine. Remember what I said, water is a follower. So glucose is what we refer to as an osmotic diuretic, which means, all right, wherever glucose goes, water wants to follow. And that's what glucose essentially does. It is going to pull water from wherever it is. So if you are secreting glucose into the filtrate, water will follow. And then it's in the tubular fluid. And then when you resorb the glucose from the tubular fluid back into your blood, water will follow the glucose. And so water will be pulled from the tubular fluid back into the blood. All right, but when you have diabetes and you're not able to resorb all of that glucose, right, from the tubular fluid, and you start to put it in the urine, then water will follow it into the urine. And that's why diabetics have to go to the bathroom all the time. And as a result of them having to pee all the time, they're going to get thirsty because they're going to try to replace that water that they've been urinating out. All right, one last concept here, and then we'll call it. All right, we're going to talk about nutrient resorption. All right, nutrient resorption, all right, is going to be when we have resorbed whatever it is, the nutrient, completely. So let's talk about glucose here. All right, so with glucose, like I mentioned before, glucose has its own transport proteins located in the plasma membrane. Okay, those are going to be these channels, all right, that are going to move glucose in and out of these cells here. So when we're looking at glucose transport in our cells, in the tubular cells here of the kidney, we have on the luminal surface, all right, our symporter mechanism, which is called a sodium glucose symporter, which means it's going to move glucose in one direction and sodium in another. And so as glucose is moving in one direction, sodium goes, all right, down its concentration gradient. And as it goes down its concentration gradient, it will start to generate kinetic energy, right? And that energy helps to operate this transport protein. So as sodium moves down its concentration gradient, goes from an area of higher concentration to lower concentration, it allows glucose to move up its concentration gradient. Anytime you're moving up your concentration gradient, that is active transport that requires energy, right? But this does so by that secondary active transport mechanism, and the energy that we generate from this, we can power the movement of glucose. Once glucose enters the cell, then we can move glucose out of the cell across the basolateral membrane, right, into the blood, right, by our uniporters. Uniporters mean that they only move one thing. In this case, it's just going to be glucose. And so glucose will go from an area of high concentration, because it's in high concentration in the cell, to an area of low concentration back into your blood. Now this process, all right, for people that don't have diabetes or what are considered to be healthy individuals, this process is pretty much going to be 100% resorption of glucose, meaning, all right, if glucose is getting filtered out of your blood and into the kidney tubules there, and it's in the tubular fluid, 100% of that glucose that's in the tubular fluid is going to move back into the blood. And that's what we're seeing here. Here's our tubular fluid. All right, so we'll see, all right, we have our sodium glucose symporter. And so sodium is in high concentration outside the cell and in low concentration inside the cell. So as sodium goes down its concentration gradient, as it's moving across, right, it is then going to cause glucose, because it's in low concentration here in the tubular fluid, but it's in high concentration inside the cell. So it has to move up its concentration gradient. Not a problem though, because the energy that we are developing from the movement of sodium across the plasma membrane, we're going to use that to fuel the movement of glucose inside the cell. Okay, so that's going to be our secondary active transport. So now we got a lot of glucose inside the cell. We want to get it back into the blood. So we have, all right, our glucose uniporter here, okay, high concentration here, low concentration in the blood. So now we're going to move glucose, all right, from inside the cell, outside the cell, and into the bloodstream here. So we go from a high glucose concentration to a low glucose concentration outside the cell, and it moves back into the blood. And that's how we resorb all of the glucose. All of our glucose will be resorbed that way. All right, and then the last one, I promise you, this is the last one. All right, proteins. All right, first of all, I mentioned before at the beginning of this lesson that proteins are normally not going to be filtered across our filtration membrane. The large size proteins will pretty much remain in the blood because the basement membrane prevents the passing. Smaller proteins are usually prevented from passing by the filtration slits, but sometimes we'll see them pass, okay? So small and medium-sized proteins can, all right, infiltrate our filtrate fluid there. All right. And in some cases, we can get some large proteins, but normally we don't expect to see that too much in people with a pretty healthy filtration membrane, but we can still see it. Point being is, all right, what we need to do is we want to return these proteins back into the blood. So we're going to move them from that tubular fluid back to the blood. How do we do that? On the luminal membrane there, okay, of those cuboidal cells, we will move the proteins either by pinocytosis, okay, that's that cell drinking, or our receptor-mediated endocytosis. Point being is both of those processes will bring the proteins inside the cell. Once they're inside the cell, okay, the lysosomes will go to work on them and break them down, or enzymes, all right, the peptidases will break them down back into amino acids, the building blocks for our proteins. And then we just dump those amino acids back into our blood, and the amino acids can circulate around and be used by cells to make more proteins, or we can use them for gluconeogenesis. There's lots that we can do with the amino acids. We don't just have to reconstruct the protein that we just broke down there in the kidney there. All right, that's it. I promise. All done.